Yeah. Yeah, with that, we may as well start, I think, if the numbers have sort of leveled off a little bit with people entering. So thank you, everyone, for coming along to the first Pesticide Watch webinar. We hope that this is the first webinar of a series of webinars that we'll put together around, pesticides, regulation around pesticides, past monitoring, all sorts of things. But this first one is really going to be an introduction into the context which Pesticide Watch sort of sits in the current monitoring landscape of pesticides, what are pesticides, that sort of thing, and how our approach is different and we hope is going to generate some really interesting data. So yeah, I just maybe quickly introduce myself. I'm Brady Hamilton. You've probably heard from me via email or on Facebook, or maybe you've seen me speak at one of the other citizen science events I've been to. I'm a PhD student at Deakin University's School of Life and Environmental Science, and Pesticide Watch came out of my honours project, which I did a couple of years ago, and it's slowly been growing since then. Some of you may have contributed to a pilot study that we did for this as a sort of proof of concept, and now we're running at full scale and hoping to get some really great baseline data around pesticides. So my background was originally in high school teaching, which I quickly abandoned after lockdowns and decided to jump into research again. And that's where I did my honors, started Pesticide Watch. And yeah, now I'd basically say my area is analytical chemistry primarily, but I'm really interested in the environmental applications of that. I'm also joined today by Jess, who you've probably heard from already in the first couple of minutes. So she's also a PhD student at the School of Life and Environmental Science at Deakin. And yeah, if you have any questions, you can pop them in the chat along the way. Jess will be monitoring that. She'll also be asking some of the prepared questions that were submitted before the talk, today. So yeah, if you have any technical issues as well, hopefully Jess will be able to get around to that. If not, I'll try to as well. So let's begin. So firstly, I'd like to acknowledge that I'm coming to you today from Wurundjeri country in Melbourne, so from Deakin Burwood campus. Pictured here is Kooyongkoot Creek, which is Gardeners Creek, which runs through the middle of our campus and is actually my sampling site. So I'd like to pay my respect to the elders past and present, and also to those who are custodians of the land that you're coming from today, and also all the sites that are part of Pesticide Watch. And I'd like to thank them for their custodianship of our aquatic systems. So first thing is community science. So primarily Pesticide Watch is a community science program, or citizen science, as it's also called. Now, citizen science data now far outstrips the amount of non-citizen science collected data in platforms like the Atlas of Living Australia, which some of you may know about. I'm sure a lot of you are involved in citizen science in one way or another. And it has a couple of key benefits. Firstly, it can increase the rate of data collection. It's a really cost-efficient way of conducting research. And it also accelerates the rate at which we can learn new things. But probably equally, if not more importantly, is that it really fosters a sense of environmental custodianship in experts and also the community, and it's also an opportunity for an exchange of knowledge between those two groups. But even though citizen science has been capitalized on pretty well in the conservation biology space, it hasn't really been tapped into so well in things like emerging contaminants. Now, we know that synthetic chemical contaminants is a growing issue, and it's actually a sharply rising issue. If you think about it, we had, as you can see in the plot there, not many potential synthetic chemical contaminants from about 1960. But now we have just a huge rise in the number of potential contaminants in our environment. And really, that's within the lifespan of my parents. So that's a pretty incredible rise in this potential risk. And unfortunately, most are unregulated. So you can see in this plot here that the number of regulated chemicals is indicated by this star down here. So only a small fraction are properly regulated. That means that a lot of these are ubiquitous in freshwater systems. So we know definitely pharmaceutical products are in virtually every aquatic system around the planet. We know that microplastics are a growing issue. And these are hugely diverse contaminants. So they're not one type of risk that they pose. There's all sorts of things. We've got pharmaceuticals, as I mentioned. personal care products. PFAS has had a lot of attention in the media recently, and pesticides also fits into that. So they're also highly mobile, meaning that just because we detect it in a particular system doesn't necessarily mean that there's a point source nearby. And unfortunately, some researchers actually estimate that the environmental impact of emerging contaminants increasing more rapidly than CO2 emissions, even though there's far less research and public knowledge around this issue. So pesticides falls within this category, and I thought I'd give a quick definition as to what pesticides are, because it's unclear depending on what you're reading. So these are just any chemical that's used either to deter or eliminate, basically kill, a living organism. They're usually categorized by what they target. So we have things like herbicides, insecticides, fungicides, and so on. They can also be categorized based on their chemistry. The interesting thing about pesticides, particularly as an environmental contaminant, is that they're designed to have direct biological effects, usually lethal effects, at very low concentrations. Also within pesticides, we're not talking about one type of chemical with one type of behavior. It's a hugely diverse group of chemistries. They vary so much in mobility, persistence, their ability to bioaccumulate, and also in their ability to be toxic. Now that image in the top right is one that I took at Bunnings not long ago, and this is a way that the community relates to pesticides, just a huge array of different pesticide products that are available just off the shelf at a place like Bunnings, from really small spray bottles right up into huge 5-liter barrels of these things. The other thing that the community tends to see are things like this, which is clearly showing some land management that's happened. A herbicide has been sprayed here, and even though there's a bit of a buffer zone, we can see that there is a stream there. So of course, a passerby is going to think, I wonder if there's a problem there. So this was actually pictured by my supervisor at Gardeners Creek on campus probably a couple of months ago while we've been doing this research. So we want to take communities' understanding and ability to engage with this issue beyond these two things. So in Australia, there's actually over 11,000 different pesticide products that are currently registered with the Australian regulator, which is the Australian Pesticides and Veterinary Medicines Authority. And usage has doubled. I also saw that this is close to tripling since the 1990s. And that's usually due to intensive agriculture and growing human population. So the context they're generally used in are things like food production, industry, and homes, so the domestic environment. Now they do enter the environment, usually via surface runoff, particularly for contemporary pesticides. Rain-driven surface runoff is probably the #1 way that these pesticides enter the environment. They can also enter via spray drift, via wind, and also leaching in from the sediment. So they do vary in their environmental persistence. So some can last just hours and they're in the system and out of the system, or they've broken down within that system to non-harmful chemicals. However, some do last decades, and I'll mention one of those in a second. Some of you probably already know what I'm talking about there. We also have a new category called pseudopersistence, which means, so this is just being acknowledged now. It's basically where a chemical contaminant doesn't necessarily have the properties that makes it persistent in an environment, but because it's applied so frequently in that area, it means that it's always present in a particular aquatic system, meaning that it sort of behaves as a persistent chemical, even though it shouldn't. So that's called pseudopersistence. And once they're actually in the environment, there's a couple of things that can happen. The first thing is that we can have acute lethal effects. So basically, we can have organisms die. And we know that this isn't just happening overseas, this is happening in Australia. So there are large biodiversity losses in macroinvertebrates in aquatic systems as a result of insecticides. So if the concentration isn't high enough to cause something like lethal effects, if the species aren't sensitive enough, then we can get these chronic sublethal effects. And these can affect organisms physiologically, structurally, interestingly, even behaviorally. And these all tend to be quite detrimental to that system. But the particular effects that we see do depend on a few things. It depend on the mode of action of the particular chemical that we're seeing, so how it works, the concentration that it exists in, the duration of exposure, and also the species sensitivity in that system. So really we need to learn these things. We need to know what particular chemicals are out there, what concentration they're in, so how much of them's there, how long they're sticking around, and what they're likely to be toxic to. So unfortunately, pesticide regulation in Australia isn't a great story. For example, DDT, an organochlorine pesticide, was actually banned about 15 years after the United States banned it. And this was well after the publication of Silent Spring, and DDT was the central pesticide that was discussed in Rachel Carson's book. So we're a bit slow off our feet, especially in that time frame. But as a result, we're still detecting these organochlorine pesticides in Australia today, and that includes the Great Barrier Reef, unfortunately. So, and other pesticides of concern internationally currently are still approved for use in Australia. Here's one example that's currently being used in Australia. It's called atrazine. And atrazine is a widely used herbicide in Australia, not quite as widely used as something like glyphosate or Roundup. But it is still one of the most widely used pesticides, and it's detected in high levels in water quality monitoring. And it has been deregistered in the European Union for, and this is a quote, ubiquitous and unpreventable water contamination. But fair enough, as I'll show you in a second, the European Union is ahead of us when it comes to banning these things. So maybe we're going to follow suit relatively quickly, except this was actually in 2003. So they deregistered over 20 years ago, saying that they just can't prevent the water contamination as a result of atrazine, and yet we're still using it widely here. It's available online if you wanted to purchase some. So this is 15 kilos you can buy for $165, probably about the price of, an expensive bag of dog food. Except it's a persistent chemical contaminant that is causing problems in our aquatic systems. And you can get this with free and fast shipping, so it's nice and convenient to be able to get hold of this sort of thing. Here's a list of other chemicals which are currently banned overseas in the European Union, but still used widely in Australia. You will have heard of some of these in the news recently, particularly paraquat, I would imagine. Diuron gets a fair bit of attention as well. Dimethoate was restricted, I believe, last year. And you will definitely have heard of neonicotinoids, which are suspected to have a role in colony collapse disorder with honeybees. That's yet to be proven or doesn't have a lot of support around it, but it was banned in the European Union because it's suspected to be involved. Here, we're still using things like imidacloprid and neonicotinoid quite widely. And if we wanted to know how much of each of these dangerous chemicals are being used, it would be quite tricky. There isn't a great register of use data for pesticides in Australia, so it's not really collected in the first place generally. But then it's also not reported, and it's certainly not reported publicly. So this is probably the best publicly available data we've got for pesticide use in Australia. It's sales data, which is published annually by the APVMA, so the pesticide regulator. And it doesn't have great resolution, because if we wanted to learn about how much atrazine is being used, well, it's buried somewhere in this figure, which just says herbicide. It tells us that it contains about 4,000 different products and how much that's worth. So there isn't great resolution. We don't have great use data, let alone exposure data. It's really difficult. However, Deakin University researchers recently, probably I think about 3 years ago, published this paper, 2021, I think, which shows an estimate based on the sales data published by the APVMA and also some Food and Agriculture Organization data and some other sort of scattered data sources to estimate where the most aquatic ecotoxicity is happening. And it highlighted some potential hotspots. However, this is sort of an estimate based on some really poor use data. So we need complementary monitoring data to go with something like this so that we actually have a good understanding of which areas are most at risk of pesticide contamination. This is where pesticide monitoring comes in. And I quickly just wanted to mention that pesticides are still designated as a contaminant of emerging concern, as I mentioned at the beginning. And it's sort of hard to understand why. We've known about the negative effects of pesticides since at least the 1960s. So I can only understand this as being an indication that it's just not something that's routinely monitored. And so we don't know much about it. So why is this the case? Well, monitoring these things is really tricky. There's no litmus test or probe-based test you can do to sort of derive a pesticide profile for a particular system. And because of that, it's expensive. You need mass spectrometry equipment. You need labs that are set up to run those instruments and people trained to do those things. So if you wanted to run some of these samples for pesticides at a commercial lab, it would cost a couple of $100s per sample. It's also time consuming. If we wanted to do a large-scale pesticide baseline monitoring study in Australia with just a single operator like myself running Pesticide Watch, or even if there were a dozen researchers working on it, to be able to get the geographical spread around Australia would just be so labor intensive that it wouldn't really be viable. That's where our project comes in. So this is Pesticide Watch, which was the idea of my supervisors, Damien Callahan and Julie Mondin, but also, myself, who sort of took the helm of this project in my honors project. And really, the aim of this program is to put together a simple, cheap, but robust pesticide monitoring program. So it does this by engaging people like you, so community samplers of freshwater systems. So you will represent school groups, community groups, even traditional owners groups. There's so many different groups involved in this program. So we've provided you with simple sampling kits, but we're pairing that with some state-of-the-art instrumentation that we're lucky enough to have in-house at Deakin to give us some really robust monitoring data. So how do we keep it cheap? Well, you've probably all received the kits that we've sent out. The materials in them are pretty inexpensive. It's basically some plasticware that helps you collect a filtered water sample. We do the analysis in-house, meaning that we're not having to pay a couple of $100s per injection onto an analytical instrument. We can do it all ourselves when we can in large batches. And I've worked hard to minimize the sample preparation here for a few reasons. Firstly, it does keep it affordable because we don't need to buy extra plasticware, extra glassware, extra solvents. It also makes it easier because the more we can automate these things and not do as much sample preparation, the less I have to be labor intensive with each sample, and that takes time. And also it's better for the environment to have, less consumable plasticware in the lab, less organic solvents having to be used for extractions. So how do we keep it simple? Well, you've probably had a go at our sampling protocol so far. It's a fairly easy filtered water sample. Yeah, there are some tricky rinses. You have to get the order right to make sure that everything's been rinsed with that native water, and some bits have to be rinsed with the filtered water. But it is a fairly straightforward sampling protocol. We haven't asked people, for example, to wade into the stream and collect us a sediment sample for this. And then these are mailed into our lab for analysis. So how do we keep the sample integrity there to make this a robust thing? Well, firstly, we want to limit the biological activity, and we do this firstly by filtering those samples, which takes out things like bacteria, which stops those pesticide compounds from being degraded in there and removes other interferences. And then also freezing further slows any degradation of that sample. So that's how we collect a high quality sample, but how we actually structure this and why? Well, we are collecting grab samples, and grab samples are different from something like passive sampling, if you know about this. So we're not collecting data constantly throughout a month where it's being, the water's being absorbed onto a surface and then we're taking that and extracting what's in there. We're taking a particular sample at a particular time, and that really just gives us a snapshot in time. And grab samples are often criticized for this. So I just quickly wanted to show you what ours is doing. So this is a representation of changing pesticide concentrations over time in a particular stream. So just a representation. A lot of grab sample studies really just grab one particular sample at a particular time and use that to represent the whole system. And obviously this isn't a great approach. Sometimes studies are limited in terms of what they can afford to do, but this doesn't provide a great representation. So our compromise is that we've asked you to collect samples repeatedly from those sites across the year, And so then we get a lot of different snapshots of what's going on. And hopefully we can get a picture, or at least an idea, about the extent of the pressures that that particular system is facing. So you'll all be collecting up to 12 samples monthly. So some of you will be collecting 6 months worth of samples. Some of you will be collecting 12 months. And we're hoping to capture that seasonal variation as a result. However, we do have to acknowledge that contaminant profiles in these systems do change on a daily, in some cases even an hourly scale, depending on how fast they're flowing, which still remains a challenge for this kind of monitoring. This is what that change over time looks like, though. This is from our pilot study that we did last year, and these are some sites along Bendigo Creek. And you can see that the cumulative pesticide concentration is represented by the radius of the circle, and then there's a bit of a traffic light system to show you the number of different pesticide detections at each site. And as you can see, no single month, no single sample really captures the extent of the pressures on that system. So we're hoping to get up to 12 of these for each site throughout this monitoring program so that we can really get an idea about, the limits of what these systems are facing. Speaking of the pilot study data, this is some of the most frequently detected pesticides that we had in that study. Now, just as a bit of a caveat on this isn't peer reviewed. It hasn't gone through the peer reviewed process, but this was part of my honors thesis. And as you can see, there are some of the usual suspects in here, things like DEET, some, of those triazine herbicides like simazine, atrazine that I mentioned earlier. The interesting thing, though, to take away from this is that I'll just quickly show you the number of pesticides which have actually been represented in routine monitoring in Australia before. It's just these ones. So these ones have been represented in more than 10 monitoring studies in Australia. So they're the ones that we're looking for. There are a lot there that they haven't been looking for. So even though they do capture a lot of what's going on at the top end there, they're missing a lot of what's going on in the middle and the end, and even at the very top there. So interestingly as well, you can tell that, so blue indicates fungicides. Fungicides are typically not represented well in monitoring, but they're represented in our study. And as you can see, those blue peaks haven't been looked for in those studies before. So we're hoping to be able to sort of elucidate what's going on with fungicides in our system for the first time. So only 9 out of 50 for those top most frequently detected pesticides have been represented in those studies. This is another quick example of the sort of detective work that we can do if we really interrogate our data set that we collect. And this is just one particular example that just happened to catch my attention as I was going through that pilot study data. It's about a fungicide that occurred at a site in Ammermoor Creek in Queensland. I don't know if anyone from Ammermoor Creek monitoring site is here today, but this is only one sample. Even from this site, we collected 6 samples from this site. Only one of them had this particular pesticide there called quintazine, and it gave a peak on our instrument, which is a high resolution mass spectrometer. So we're pretty sure that's right. And when we spiked some purchased quintazine into some pure water and also ran that on the instrument at different concentrations, you can see that the instrument was performing quite well. So we've got some good linearity there. The instrument seems to be measuring that quite well. Now, there was another pesticide that was detected in only this sample and none of the other ones, and it's called pentachloroanisole. And that's actually a degradation product of quintazine. When we spiked some of that into some ultra pure water, we could see that across a concentration range, the instrument was performing quite well for that as well. So we're pretty sure that's what we were seeing. When I looked up on the APVMA website what this quintazine compound was registered for, 2 of the 5 that uses for it were related to peanut cultivation. And so just out of curiosity, I had a look at where Ammermoor Creek is versus where peanuts are grown in Australia typically, and there was a pretty good correlation to that area. So this doesn't necessarily say anything. We haven't gone back and verified this or looked at point sources, but it's just interesting to see the kind of detective work that we might be able to do to sort of tell some stories about the pressures that certain streams are facing in Australia. And we hope to do this sort of detective work when we have our full data set from this 2024-2025 program. So let's talk about that program. Firstly, I wanted to mention pesticide monitoring and the targets that we're looking for. So pesticide targets, I just mean that's what we've set the instrument up to look for. And these are three of the probably biggest scale pesticide monitoring programs that we've had in Australia. The first one, Reef 2050, was done by the Queensland state government. The pesticide water monitoring program was in Tasmania, and pesticide detectives was run by RMIT. So as you can see, pesticide detectives was doing quite well. They were looking for quite a lot of pesticides, about 110. However, our program looks for, well, can measure quite a few more than that. So we can measure close to 400 different pesticides. And that number is growing as we get our hands on more analytical standards to make up some standard mixes to be able to measure these things. If you have a look, though, at what we can actually just screen for in total, so not just measure, we just do a screening, we can see nearly 800 different pesticides. So we get a really good coverage of the different pesticides that are likely to be being problematic for our aquatic systems. And when you take into account as well that 2 of these programs are no longer running, then you can see that we're contributing quite a lot to Australian pesticide monitoring at the moment. I will come back to Reef 2050 though at the end of this presentation because I think they're doing some amazing things, which I would like to emulate at the end of this project. So this is the extent of the sampling campaign. You may have already seen this post on the Facebook page, but these are the sites that we've managed to get together. And I'm so grateful that you've all been able to contribute to this program and that there's been such ready uptake of something like Pesticide Watch. I didn't really know whether this would be the case when we first started the pilot study, but it's great to see that there's so much appetite to learn what sort of emerging contaminants are in your environment and how that's affecting your aquatic systems. So we managed to get some sites in every state and territory except for the ACT. We were so close to getting all of them, but these sites all represent different pressures. So we have agricultural systems, we've got some pristine sites, so wilderness and forested areas, we've got sensitive systems like wetlands, and then we've got some really disturbed sites, things like the urban fringe and the urban environment and industrial. So we're hoping that we can see some really interesting relationships with land use and pesticide profiles. So just thank all these groups again for contributing. We've got government, catchment management authorities, schools, land care, traditional owners, even universities. So really glad that we've been able to get 136 sites together. And if we look at where our sites are relative to that estimate that was done by Deakin University a few years ago, we can see that we're getting some of the hotspots there. Even in Tasmania, we're able to get that sort of band that goes along the east. So we're hoping that we can get, we can maybe do some risk assessment for some of these sites that are most at risk. This is where we are in terms of the number of samples we've received so far. So for this first sample batch, we've nearly received half already, which is great. And I know that some of you will have started a month later than the others. So you may still be collecting your third sample before you send that batch of 3 back. But yeah, we've got a great number so far. The freezer's filling up very quickly. Might need to look at purchasing another freezer or finding some more freezer space. But yes, thank you to everyone who's been able to get these in. It's been really amazing to, open these up. It's like Christmas every time they arrive and, log them, put them in the freezer. It's just great to know that they're ready to go for analysis. And this is what the freezer looked like probably a week ago. There's quite a few more than that now. And these are coming from right around Australia, which is really impressive. Now, once we actually have our analytical data, we want to find a way to share our data publicly. And I think a really great example is given by the Queensland state government with their Reef 2050 program. So this is the pesticide reporting portal. You can access this online afterwards if you'd like to. You just type in pesticide reporting portal and it takes you to something that looks like this. It's an interactive map and it shows their monitoring area, their monitoring sites. And you can select a particular site and you can choose from their list of, I think it was 25 pesticides that they analyzed for. I chose imidacloprid here, one of those neonicotinoid pesticides. And you can track the measured value of each pesticide over the monitoring program. So you can see here that they measured above their limit of detection. Most of the time, it looks like the levels are pretty low. But they've also indicated with this yellow line here, the environmental threshold there, above which you would expect toxic effects to occur. And there was an exceedance here. But I think that this is a great way to be able to communicate data to the community. And this is what we would aim to do. However, I'd love to have this across all of Australia, not just this area of Queensland. And yeah, I think a good example of this is provided by the Netherlands. So they do great pesticide monitoring. They carried out these structured surveys via the water boards, which I imagine are similar to catchments. And they not only report pesticide concentrations, but they also interpret this for the public. So in terms of long-term trends, links with different types of land use and exceedances of certain thresholds. They publicly publish this data in a pesticide atlas, which I'll quickly show you now. I apologize for the Google Translate with this one. Unfortunately, it doesn't work perfectly. For example, I'm pretty sure dust here is supposed to be residue. So you can toggle between different years. You can toggle between different pesticides. You can filter by different land uses, all sorts of things. And you can just click on each dot, each sampling site, and find out what measured values there are. So this is something that we would love to see for Pesticide Watch once we do have all our analytical data. And we're aiming to start hosting a website very soon. So this will be uploaded to that website. Of course, I've been talking a lot about pesticides. We know that tracking waterway health is, it contains a lot of different pressures there. So we are actually, though, through this program, getting quite a lot of pieces of the puzzle. So it's not perfect, but we are looking at trace elements as well. So we'll be able to estimate the risk for those systems as a result of things like metals. And we are going to be looking at pesticide risk as well. And then also, because you're doing your water quality monitoring already, we'll be able to get some of these environmental parameters as well, things like temperature, pH, sediment load, sometimes nutrients as well. So we can potentially look at developing a water quality score for this. And if we build these three components into an environmental water quality index, then this can give us a single number, which will help indicate the health of that system as a result of, water quality, metals, and pesticides. And that can help managers and community track changes over time in a sort of really approachable way, something that can even be communicated in things like the classroom for any teachers that are here. And it's no small thing that this is a community approach. So this isn't just, as we see it, a sampling campaign, it's also like an education campaign. I think that the public has sort of been left out of the loop when it comes to this environmental issue. So we want the community to learn things, and we also want to learn things as well. And it's about exchanging that information between us. So I think we have a lot of shared questions here. We want to know how pesticides are actually used. Are they actually ecologically harmful in the levels that are actually, exposing aquatic organisms? Have they been regulated appropriately? Are there any that have sort of been overlooked that nobody's really monitored for before? And importantly, developing these pesticide profiles, so what pesticides are actually in the waterways. So yeah, as I said, this is a sampling and an education campaign, and we do really want to continue these webinars. So webinars, emails, social media, we'll get that website up and running very soon. I'm just getting approval for that now. And also we'd like to distribute some questionnaires. I can talk about this down the track, but we'd love to learn about things like how you perceive pesticides. Do you perceive them as risky or relatively safe? What's your relationship with pesticides? All these sorts of things. It's hugely valuable data. I'll talk more about those questionnaires down the track. So this has been sort of an introduction into Pesticide Watch and what we hope to achieve in the context that we sit within. But I was thinking, for the future webinars, I'm hoping that you might contribute to some of these topics that we end up exploring. So these are just some suggestions that I thought might be good. So for example, what we do in the laboratory here, what past pesticide monitoring in Australia has revealed and how they structured their surveys, maybe the process of pesticide approvals and regulation in Australia, and things like observe the impacts of pesticides on Australian streams from the literature that already exists. So if you have any questions or suggestions about what we might be able to do in a future webinar, feel free to chuck that in the chat now, or you can email me, which is available on the Facebook page, or you can just directly message me on that Facebook page as well. So yeah, I'd love to have your input. Maybe there's something I haven't thought of that would be great to cover. So with that, I'd like to thank everyone for their attention. I've just been talking for probably half an hour non-stop, so I'm sorry about that. But I'd like to also just quickly thank Eurofins as well, a commercial lab that's helped us with some of the method validation for this, and also the CSIRO, who's taken me on as a student affiliate and lets me use some of their cool gas chromatography instrumentation that we don't have at Deakin at the moment. So thank you everyone for your attention. And yeah, I think we might have a look at some of those questions now, Jess.
Yeah, we've actually got quite a few really good questions. I'm not sure if you
Okay.
can see the chat, Brady.
Let me get
I
that
can
up.
start reading a couple of them. So from Anthony, we have the question, can the plastic sample tube used in the sample or the sample collection sort of leak into the samples? Are the sample tubes fluorinated?
The sample tubes are polypropylene, so they themselves aren't fluorinated as far as I know. But we use something called triple quadrupole mass spectrometry, which tends to help us overlook things like plasticizers, which leach into the sample. I think we wouldn't do something like microplastics analysis on these samples because we are collecting using plastic, so that would be out of the question. But yeah, I think the main issue we're having with the plastic tubes are some of our more hydrophobic pesticides tend to be clinging to the sample tube. So we're trying to find a way to sort of desorb those from the surface. However, for most of the pesticides, almost all of them, we're not seeing any interference. We're getting nice clear peaks. So yeah, hopefully that answers that one.
Yep, that sounds good. And we had a question from Julia about the specific pesticides that were sort of seen in the pilot studies. So
Yeah,
there were a few concerning ones that you were mentioning.
yeah. Yeah, in terms of risk, we haven't really. I mean, we did sort of compare some of these to their environmental threshold values, and there were only a couple of exceedances of those in Australia for that pilot study. Um, so, the specified limit depends how happy you are with those limits, whether they're appropriate or not. But we didn't see many exceedances of the safe limits. But the fact that we're still detecting them so frequently is probably an issue in themselves. But yeah, we didn't see any of those sort of organochlorine pesticides, those things like DDT or DDE or dieldrin. So those are probably the more toxic ones. But yeah, these contemporary pesticides did pop up a fair bit.
Yeah, very interesting. The next question we have is there an opportunity to join new sites to the program now?
Um, yeah, that might be one that's worth an email. I'll need to talk to a few people. Um, our resources, so we don't have any spare kits at the moment. Potentially, I can't promise anything, but we can try to see what we can do. We are a few months into the program now, so we still would want 12 months so that we can sort of do a risk assessment based on 12 months of samples potentially. But yeah, feel free to send me an email. Hopefully my email is available on that Facebook page.
Yeah, I can also share your email in the chat here as well, Brady.
Oh, great. Thanks for that.
Our next question is, it would be interesting to also include domestic water supplies, particularly in areas that don't have any sampling, things like rainwater tanks as well. Would that be within the scope of this project?
Not currently, but if we do expand the project even further, I think that's a great idea as well. Yeah, currently we're just looking at environmental water, and that's because we have, committed volunteers who are currently doing water quality monitoring there. So this is sort of like an additional thing we're offering those people. But yeah, I can see if this attracts a bit more funding, if this can become an ongoing project. Why not? We can probably do all sorts of things. Farm dams, would be a great one as well. So I think there's all sorts of different sources of water that would be interesting.
Yeah, great. And next we have a question about what the role of riparian vegetation and buffers upstream of the sites in monitoring the pesticide mobility into the waterways.
Yeah, I mean, that's not something we've really considered. Yeah, in terms of reducing pesticide load or slowing pesticide load, I'm not really sure. There's probably something in the literature. I can have a look into that and see if there's anything there, but yeah, I don't currently have a clear answer for that one at the moment.
Yeah. And what do we know about the sensitivity of particular species of macroinvertebrates to different toxins?
It does vary quite a lot. Australia doesn't have a huge number of um, environmental thresholds for the suite of pesticides which are actually being used in Australia. So we fall well short of actually having official numbers on, how toxic this is to certain organisms. But there is a lot of toxicity data that's available on big databases. Part of this project is going to be trying to pull all that toxicity data, which is usually done either to macroinvertebrates, crustaceans, fish, and try to bring that into an estimate of risk for each pesticide to each type of organism, and then try to estimate the risk at each site based on all of that. So it's going to be quite an intensive process, but yeah, basically toxicity data is very varied, and it doesn't really cover a huge amount in Australia currently.
Awesome. Yeah, thanks for that. We have another it's a bit of a specific question. Associate Professor Matt Landos. So, have you explored the effect of pre-filtration on pesticide detection as a result of many chemicals having high KOW driving their affinity to sediment?
Driving their affinity to sediment. Um, I have looked at pre-filtration in terms of the different syringe filters we're using. I hope that that's the question that you mean. So I looked at a bunch of different filters and had a look at recoveries just from spiked samples. And we're only going to be measuring the ones that do have acceptable recoveries in a sort of 70% to 120% recovery range. But yeah, and I think I mean, those high log KOH or KOWs, so the more hydrophobic ones tended to be more of a problem with the sampling vials rather than the syringe filters. So yeah, I hope that sort of answers the question. But yeah, I'm sort of maybe I need to know a bit more about the sediment that you mean. So maybe yeah, feel free to send me an email as well, and I'll try to answer that more clearly.
Yep, that sounds good. Um, so yeah, we have another question talking about some pesticides having a short lifespan in water compared to when attached to sediment. Are you considering including sediment in your study as well
I'd
as
love to. Yeah, I think that'd be great. But as I mentioned, we sort of felt we wanted to make this a really cheap and easy monitoring program to run. And I think what's available in the water is probably what's likely driving a lot of risk for organisms in there. So that's a bit of a compromise that we've done. I'd love to be able to eventually pair this with sediment samples, though. Um, I can understand that would be a more labor intensive and expensive way to do this sampling, but it would give us a much fuller picture. In fact, I'd actually love to be able to do grab samples like we're doing, sediment samples, and passive sampling at targeted sites. That'd be the dream of getting pesticide monitoring in each of these sites. So, yeah, hopefully in the future.
yeah, there's a lot of room to add to this sort of project, isn't there? So many
Yeah, definitely.
possibilities. We've also had another comment that it would be interesting to understand the bioaccumulation of pesticides in higher predators, like such as the platypus.
Yeah, yeah, having, being an insectivore, that's probably a big issue. I'd love to be able to do that as well. We're not currently looking at extracting pesticides out of organisms, but this is something that someone mentioned to me last week when we were talking about my project. They mentioned could do macroinvertebrate sampling and look at the diversity and extract, pesticides out of those and see what's actually accumulating in them, which, would be great for an exposure assessment, because then you're actually seeing directly what is going to be a problem for certain macroinvertebrates. But yeah, higher predators as well, probably complicated, but, why not? That sounds really interesting. But this is probably more for the future and probably outside of just environmental monitoring at that point.
I think that might be all of the questions we've got in the chat for the moment. Did you want me to read out some of the questions that were submitted before the session, Brady?
Yeah. So there were some that were submitted, I think, from Merry Creek Paddle Group.
Yeah.
And one from Cornish College, I think.
Perfect. So the first question we have from Merry Creek Paddle Group is, when will the samplers get the results, and how will they get the results?
Well, I think we'll probably be able to get results together about 6 months after we receive the last samples. So I think the last samples will be collected in July or August, depending on when a group started. So that'll be next year. And so probably about 6 months after that, by the time that we do the, analytical stuff, then we look at the data analysis, make sure that all the checks and balances have been made. This is quite sensitive data at the end of the day. So I think about 6 months is about right. And it'll be communicated. Firstly, we'll be aiming for a peer-reviewed paper. Once we're comfortable with our data, we'll then be doing webinars. So probably a summary webinar about what happened in the whole network. And then if people want specific answers about their sites, we can maybe set up smaller meetings for that as well.
And will there be someone to help explain the results once they're available?
Yep, I'll be there. Yep, every step of the way. Don't worry.
How many pesticides are you testing?
Well, we're able to measure, as I said, sort of about 400 different pesticides across 2 different instruments. So one at Deakin and one at CSIRO. But we can actually screen for close to 800. And one of our instruments can actually potentially do untargeted analysis, meaning we can potentially pick up things that we're not necessarily looking for. So that could add another layer. I won't guarantee that that's the road that I'll go down, because it is on a short time frame. But the targeted approach means we can screen for close to 800.
Awesome. And is there access to further testing into 2025 and 2026? I
hope so. It's a big question. That depends on whether we can attract, the appropriate funding and collaborators. I hope it does. I think that this is such a valuable program, and I've just come back from a conference in Canberra, and it was an ecotoxicology conference, and everyone was stressing the importance of bringing the community along for this kind of monitoring. And this should be done more. So hopefully that's reflected in people's appetite to help out and collaborate with a program like this.
Yeah, absolutely. And you mentioned that you're hoping to have a peer-reviewed paper come out of this project. What else will you do with the results?
So yeah, when we if we do see exceedances, we'll have to chase that up. But I think firstly, we'll be communicating to you. You're the people that collected the samples. We'll then be looking at communicating to the catchments if we think that some management intervention might be required. And also, the catchments have helped us coordinate this program quite a lot. And yeah, if we do see exceedances and identify a risk, then we will be chasing that up as well to make sure that our results are verified with interlaboratory studies. And then once we're sure there's an issue that is providing an acute risk to people or the environment, we'll then be chasing that up even further with the regulator.
We've also just had another question put into the chat. Do you have a report or presentation outlining the results from the pilot study?
Yeah, I do. I can send one out to you. I think I've got your email, Simon, so I can probably just send that out to you if you like. I'll just make a note. Yeah. Yeah, great.
So then,
I'll send that out after this.
We also have a couple of questions from the Year 5 students at Cornish College. So from Toby, Toby's asked, asked if we could use something else that serves the same use as pesticides.
I think that we can afford to be more creative with how we address things like pests, rather than applying, these synthetic chemicals which are designed to kill things at low doses. Um, and the way we apply them also just seems, a bit excessive. I hope that we can have a more targeted approach and try to be more creative in terms of our approaches. I mean, even Rachel Carson, in a book in the 1960s, was outlining ways that we can biologically control for pests rather than using chemicals. And I don't think that we can stop using pesticides. We have a growing human population. We're very reliant on these things. But I think we can reduce it. We can try to make it more targeted, and we can try to stop it getting into the environment.
And can a low amount of pesticides be good for waterways?
I'd probably say no. I think, these are systems which are, complex. They've been around for, they're the result of evolution over thousands of years. Um, and though most of that time was without any synthetic chemical contaminants at all. So this is a new pressure. As I showed, in the 1960s, these things were brand new. So yeah, I don't think that a small amount will be good for these systems. Yeah, I think we need to try to remove any exposure to not only pesticides, but things like pharmaceutical pollution, all sorts of things in these systems. They're just a new pressure, which is putting a lot of stress on them.
We've also had a few more people asking about the pilot study. I'm just wondering if you would be able to maybe make it available to more people,
Yeah, I
or
can send that out to the mailing list. If you're not part of the mailing list, please get in touch. So I think I've got about maybe 60 people on that mailing list. But yeah, just send me an email and I'll add you, and then you can just be in the loop. And I'll send out a new email with that pilot study report.
fantastic. Thanks for that, Brady. Our next question is, do water transplants clear out pesticides?
Yeah, so if this is about adding new flows, like excess flows to the system, then that's probably, it's a bit of a fallacy called the solution to pollution is dilution. I don't think that's necessarily true, because these things can have negative effects at such trace concentrations that, just trying to flush them out or dilute them isn't really going to do anything. And if they've already been taken up by organisms and then other organisms are consuming those organisms, I think we can see these things concentrating in the environment anyway. So I don't think that's a quick solution to this.
Our next question is from Tom. What is a pesticide's lifespan?
Yeah, so that can vary. So as I said, some of these break down really quickly in the environment to, relatively harmless things. Some of them are just, they take decades. We all have DDT in our blood, very likely. And this has not been used in Australia since the 80s, but it was just used so much. It's so persistent in not only the environment, but in organisms, that we all likely have things like DDT in our blood. So yeah, they vary quite a lot in terms of how persistent they can be and also how toxic they can be.
And what animals do pesticides affect?
That would depend on the particular pesticide and its mode of action and what it particularly targets. But I think it's helpful to think about these environments as systems, and if it affects anything in that system, then it's likely going to have a detrimental effect to the system overall, and just place stress on that system. So yeah, it's not necessarily about one pesticide targeting one organism and causing problems. It's a system. We also know that there's a cocktail of different chemicals in these systems that exist currently. So it's their interaction as well. It's a really complex, situation out there.
And can we replace pesticides with a safer alternative? I know you mentioned sort of trying to limit our use, but yeah, what are your thoughts on that?
I hope so. I know that some of the pesticides which are causing the most problems, it'd be great if we know that they have toxic effects to be able to find a comparatively safe version that does the same thing, or a safer version that does the same thing. That's not always necessarily possible. But like I say, I think we could just be more creative with how we do pest control and try to avoid using synthetic chemicals where we can, even if that means more effort.
And when did pesticides start to cause a problem? Do you think?
Immediately. So these synthetic pesticides generally started being used after World War II, which was, within the lifespan of my grandparents, since they started being used. So it's still relatively recent. And they started causing problems almost immediately, enough that someone like Rachel Carson wrote her book, Silent Spring, to make the public aware, sort of raise the alarm bell about these toxic chemicals and how toxic they were and how inappropriately they were being used. So yeah, almost immediately. And I think it's just a sharply growing issue, even though there is some acknowledgement that they are dangerous now.
And which state in Australia have you seen being most affected by pesticides?
Interestingly, from the pilot study, that it was pretty even across all the states and territories, except Tasmania was slightly better off in terms of the number of pesticides per sample that we saw. And they also had 4 of the 6 sites that had no quantifiable detections of pesticides in them. So I don't know whether that's due to, less intensive agriculture potentially, or a lower human population. But yeah, that's what the data has shown so far. But it'll be interesting to see if that's reflected in this program as well.
Yeah, absolutely. And how can we stop the use of pesticides?
Yeah, so like I said, I think we'll struggle to completely stop the use of pesticides. We're so reliant on them now in terms of how agriculture operates, and also how we use them in our day-to-day lives. But I think we can reduce them. So that's sort of my idea, is that we can stop them getting to the environment and reduce how we're using it as well.
Yeah, absolutely. And I guess it will help being able to show what's really there as well.
Yeah, that's right. Yeah, publicly available data, I think, is a big solution to this as well, just to grow public awareness around this issue.
Yeah, so obviously identifying what is really there is the first step, but what do you do once you find a bad pesticide in a waterway?
Yeah, so this would probably be the same approach that I mentioned before. We'd like to firstly check that it does exceed a threshold, whether it's one that's specified by Australia or one that we've derived from other toxicity data. If we're sure it's a problem, then we'll be communicating to you, to the catchments, and also to the EPA to try to get some way of mitigating the effects of that pesticide getting into the environment. It might be a point source problem. It might be a dumping event. It could be anything that's actually the source of that problem. So yeah, that's what we would do. We'd try to chase this up and make sure that it's not a problem.
Yeah, that is, yeah, sounds good. We have one last question.
Yeah.
How much damage does a pesticide do compared to a herbicide?
Yep. So if we mean insecticides versus herbicides, so both are under the umbrella of pesticides.
Mhm.
Insecticides do tend to have more obvious toxic effects to things like, macroinvertebrates. But herbicides are used so ubiquitously in so many different contexts that I think it then can become quite a problem. So yeah, I think insecticides tend to be seen as more toxic and are more toxic. But yeah, I think herbicides are used so much in so many different environments that it's an issue in itself that they're so frequently detected. Yeah.
Great. Does anyone else have any other questions that they want to put into the chat? Feel free to add anything else. I think that might be all.
All right. Great. All right. Well, thank you everyone for coming along to this first webinar. I will send out some details once we've decided on a topic for the next webinar. But thank you so much for sending in these samples and showing some interest. And yeah, I'll see you at the next one. Thanks, everyone.